friends, this is Dr. Sachidanand Singh from Faculty of Biotechnology, Sankalchand Patel University. In our previous lectures, we have understood about protein structure prediction and modeling. In our previous lecture, we have also seen the understanding of how a protein structure can be predicted, modeling can be done, and what are the different modeling approaches and evaluation techniques. Now, in the current lecture, we will be understanding about other different bioinformatics softwares and other parameters which can be utilized for protein structure prediction. So, today's lecture will be on bioinformatics softwares, though on basis of other parameters like loop modeling. So, let us start. There are several software tools and packages available for protein modeling, each with its own set of features and capabilities. Here are list of some popular protein modeling softwares. As we have already seen, Modeler. Modeler is a widely used software for comparative protein modeling. It builds 3D models of protein structures by aligning with the target sequence to one or more template structure, and these structures are from Protein Data Bank, which is known as PDB. Another software is Swiss Model. Swiss Model is an automated protein structure for homology modeling server. It allows user to generate 3D protein models based on known structures in the PDB and provides a user friendly web interface. The third software to be discussed here is like Rosetta. Rosetta is a comprehensive suite of software tools for protein structure prediction. Protein design and protein-protein docking can be performed. It's particularly known for its accuracy in protein structure refinement and ab initio modeling. So, it takes up that approach. The another type of approach is multiple template modeling and loop modeling are the advanced techniques in structural bioinformatics used to predict the three-dimensional structures of proteins. So, now let us briefly introduce each one of them. So, the very first one in this lecture series we will be talking about multiple template modeling. So, let us see the definition. What is multiple template modeling is? Multiple template modeling or multiple template comparative modeling is a computational method used to predict the 3D structure of a protein by aligning it to multiple experimentally determined protein structures. These protein structures are also known as templates, and then combining the information from these templates to generate a specific protein model. So, let us see now how it works. In multiple template modeling, a target proteins, that is, an amino acid sequence, is aligned with the sequence of multiple known protein structures. They can be retrieved from different protein databases as well. They are similar to the target also. These templates can come from various sources such as Protein Data Bank, which is also known as PDB. The idea is to take advantage of the structural information from multiple templates and to create a more accurate and a model fit protein structure. So, now let us see what is the advantage of multiple template modeling is. Multiple template modeling can often result in more accurate models than single template modeling, because it incorporates a broader range of structural information. It particularly useful when a single template that does not provide a high resolution or high quality match for the entire protein sequence, right. So, we saw multiple template modeling advantage and challenges. Now, let us see what do you understand by loop modeling. Now, what is loop modeling or what is loop? It is a secondary structure, as we saw about alpha helix, beta sheets, turns and coils. So, to understand loop modeling, let us see what is loop modeling. So, what is the definition of loop modeling? Loop modeling is a subset of protein structure prediction that focuses on prediction the conformation of flexible regions or the loops within a protein structure. Loops are segments of a protein chain that can adopt multiple conformations. Segment of the protein chain that can adopt multiple conformations and are often challenging to predict accurately. That's what it's a flexible area which needs to be predicted accurately. So let's see why it is important. Loops play a crucial role in protein function. Very important aspect, protein function, as they often form active sites, binding interfaces or regions involved in protein-protein interactions. Accurate loop modeling is essential for understanding the functional and structural aspects of protein as well. So what we understood is the definition for loop modeling. Now let's see what are the different methods of loop modeling. Loop modeling methods use a combination of techniques such as, the first one is fragment based modeling, second is energy minimization, third is optimization algorithms, and for using and implying these method, it is used to predict the most likely conformation of a loop region. These methods consider factors like steric clashes, hydrogen bonding, and the very important one is van der Waals interactions. So, understanding this, now let us see what are the application of loop modeling. So, loop modeling is a commonly used in, the very important is protein structure refinement, right. Second one is protein-protein interaction studies. Third one is drug design. So, these are the three very important approach: protein-protein interaction, drug design, and protein structure refinement. It can also be applied to homology modeling and to refine loop regions in a predicted protein structure. Now, both multi-template modeling and loop modelings are valuable tools in structural bioinformatics. So, contributing to our understanding of protein structures and their functions, they are particularly useful when experimental structures are unavailable. So, now let us see, both multiple template modeling and loop modeling are valuable tools in structural bioinformatics. Contributing to our understanding of protein structure and their functions, they are particularly useful when experimental structures are unavailable or when dealing with regions of protein that exhibit structural flexibility. So, there are two approaches to use loop modeling is, what we saw is when experimental structures are not there and when structural flexibility is high. So, now let us see the very important point, what are loops? So, loops are key components of protein structures involved in many biological functions. Due to their conformational variability, the structural investigation of loop is difficult topic requiring a combination of experimental and computational method. So, let us highlight this word, the conformational variability and flexibility, that is what the loop concept came around. So, loops are protein fragments with an irregular structure connecting two secondary structure elements, typically alpha helix and beta sheet. So, in alpha helix and beta sheet, a loop is attached and are main factor in many biological functions. They are often located at the surface of globular proteins and their conformational versatility, which plays key role in process such as molecular recognition, allosteric regulation or signaling. On average, loops correspond to about 1/3 of a protein sequence and up to 1/2 of the residue of enzyme activity, active sites where they are located in. So, now let us see the loop energy landscape. So, to understand the energy landscape, it is better to understand the dynamic nature of the loop, how it performs and what is its activity. A suitable representation of their conformational space is based on the concept of energy landscape. Although one loop can exist in multiple conformations, they are not equally likely to be adopted. More precisely, the probability of each possible state depends on its associated free energy or Gibbs free energy. The energy landscape provides an overall view by displaying the energy at each point of the conformational space. The topology of this landscape gives insight into the main conformational and dynamic properties of the loop. So, to see the properties of the loop, the basics constitute behind is metastable conformations, while a saddle regions are associated with these transition states of the loop. Another method of loop understanding is scoring methods, that are another key ingredient required to evaluate the quality of the sample loop states. Scoring methods can also be classified into different methods known as knowledge based, physical based, which are also known as generally based on molecular mechanic approaches, not quantum physics models, and specifically hybrid methods. As before, knowledge based scoring functions typically linked to a coarse grain representation of the loop are the most computationally efficient alternative. However, their implicit simplifications prevent a faithful representation of the rugged energy surface of all atom conformational space. Conversely, the computational cost of detailed physics based energy functions prevents a thorough exploration of the space. Hybrid approaches try to balance the best of these two worlds. A common practices which is used as less accurate scoring function in an initial search and an atomistic detailed potential to refine only the high scoring loop states. Hence, a suitable combination of sampling, protocols and scoring function is the major burden for loop modeling, and the key features for further progress in the field is reported. In addition to the aforementioned loop modeling methods, Monte Carlo, which is also known as MC, and molecular dynamics, popularly known as MD, simulations can be used to investigate thermodynamic and kinetic properties of loops. Note, there should be a note, however, that in this context Monte Carlo derived approaches are mainly employed as a sampling engine within more sophisticated loop prediction techniques. It has been majorly reported in n number of publications about this concept. Enhanced molecular dynamic methods are suitable tools to study loop dynamics modeling. For example, MD simulation coupled with higher level representation such as Markov state models are a relevant approach to investigate the role of loop conformational changes in enzyme catalysis. Nevertheless, due to their very high computational cost, molecular dynamic simulations are often only applied in a post processing stage to refine solutions provided by much faster loop sampling methods, as reported in loop modeling sentences. So, this was all about loop modeling concept, application and principle. Why we study loop modeling, what are the applications, what are the advantages and what are the challenges. So, loop modeling in Modeler SALI lab is a separate program by which we can perform loop modeling for protein structure prediction. Loop modeling enhancement also increases the structure optimization and perfect modeling. Scientists during their loop modeling approaches, utilization is done when an unknown target sequence is been retrieved and homology modeling or ab initio modeling or threading is been performed. Later, after doing a Ramachandran plot validation, if the protein structure is not good, again loop modeling concept can be introduced to have better refinement processes. So, there is the application to get better optimized protein structure to be predicted for better scientific understanding. So, this was all about loop modeling concept, principles and application. Now, let us see some specific database or websites which performs loop modeling. One among them is DAR-AS loop modeling approach. This DAR-AS loop modeling approach is a significant loop modeling website tool to understand this. So, let us see this concept. DAR-AS loop modeling is an accurate loop modeling using fragments from remote or unrelated protein databases. Despite efforts during the past decades, loop modeling remains a difficult part of protein structure modeling. Several approaches have been developed in the framework of crystal structures. However, for homology models, the modeling of loops is still far from being solved. Under this database, it is a data-based approach that identifies loop candidates, mining the complete set of experimental structures available in the protein data bank. So, it actually takes up the help of PDB database. PDB database is maintained by Rutgers University. Candidates filtering relies on local conformation, profile comparison together with physicochemical scoring. Hence, applied to 3 different template based test sets, DAR-AS loop shows significant increase in the number of high accuracy loops and significant enhancement for modeling long loops. So, there are both approaches, short loop modeling and long loop modeling also. For all these concepts, the enhancement is been done under this web server. DAR-AS loop is a web server for remodeling of loops in homology modeling. So, the concept which it takes it up is a homology based modeling. It follows a data-based approach search method, identifying loop candidates by mining the complete set of experimental structures available in the protein data bank, which is in short known as PDB. Candidate loops are filtered based on the sequence and then ranked using the local conformation profile, and the structure fit is been obtained. DAR-AS loop database returns then 10 loop models. So, specifically as per the criteria, it gives 10 loop model for specific consent for each individual loop region. So, suppose I have a unknown sequence for which this loop modeling is been done, and if I have 3 loop regions, how many structures will be generated? 3 into 10, 30 loops. DAR-AS loop requires an initial homology model and can be used for either loop modeling or the next one is loop remodeling. For a single loop region, the results are identical. It identifies identical amino acids. For multiple loop regions, remodeling typically gives more accurate results. A special advantage is that this method proposes a prediction confidence score that correlates well with the expected accuracy of the loops. So, it takes up the scoring method approach. Strikingly, over 50% of the successful loop models are derived from unrelated proteins, indicating that fragments under similar constants tend to adopt similar structure beyond mere homology usage. So, it takes up the usage of homology modeling. This database is used to either do loop modeling or loop remodeling as well, as we studied before also. In both the cases, DAR-AS loop requires an input structure and an input sequence. So, same like modeler, input unknown sequence and an input structure as a template as we identify manually or through programmatically. If you remember in my previous videos, manually we have to go in UniProt and PDB database to understand the best structure aligned for my unknown sequence, or through the modeling or through the Python script also we can get it up. So, For loop modeling, the input structure is also required and input sequence is also required. For loop modeling, the input structure may be a gapped PDB, whereas the input sequence must contain the full protein sequence so that the query coverage can be identified. For loop modeling, it is the reverse. The input structure must contain a complete initial homology model, whereas the input sequence must be gapped, indicating the loop regions that will be remodeled. In general, best results are obtained if a remodeled loop is flanked by helix or strands. If this is not the case, you or me might consider the extend loop regions towards the nearest helix or strand. Typical runs require on the order of 1 hour depending on server load and by understanding, as it is a server based database online tool. So, it requires to give an input of structure and sequence and to initiate the process. So, after input we have to click on modeling mode. This indicates whether loop modeling or loop remodeling is to be performed. So, we can click on specific loop modeling, or we want to refine, we can go for loop remodeling. For a single loop, the results are identical, but multiple loops, loop remodeling is typically more accurate. It is also possible to do advanced loop modeling. So, after loop modeling, remodeling, there is another approach known as advanced loop modeling, which is causing all the loops to be modeled completely and independently. This is more accurate than standard modeling mode, but typically still less accurate than remodeling mode. However, each result will contain the coordinates of only one loop with gaps for all other loops. Now, let us see what protein structure we receive. Darius loop requires an initial homology model to remodel the loops. The input file must be in PDB format. It must contain only the 20 standard amino acids. Homology models can be obtained from example Swiss model server or Modbase database. So, there are different databases also discussed from where we can get the input or output. So, for getting the structure, Swiss model server or Modbase databases can be retrieved for PDB structures for the remodeling. Darius loop specializes in remodeling of the loops in homology models. While it is possible to upload experimentally solved structures, the Darius loop protocol has not been optimized for this. In addition, it is to be noted that experimental structures often contain extra atom, that is water molecules or cofactors or etc., which are not accepted by this upset database or server and must be manually removed. So, this also in the previous lecture we have discussed that open a PDB file in text file and down all the extra atoms as cofactors and water molecules can be removed before uploading in Darius loop. In loop remodeling mode, the homology model file can be uploaded directly. In loop modeling mode, the coordinates of the loop regions that are to be modeled should be removed from the file. Now, let us see what to be done with the protein sequence. In protein sequence, for the loop modeling mode, this is the full sequence of the protein that is to be modeled. It may contain only standard amino acids. In loop remodeling mode, the sequence of the loop regions that are to be remodeled should be replaced by gaps. So, this dash in inverted comma are known as gaps, and lowercase amino acid letters are recognized as gaps. So, only capital letters will be considered as amino acid, dash lowercase amino acids will be considered as gaps. In all cases, the protein structure and the protein sequence must follow exactly the same numbering scheme. For example, if the protein has residue ALA 16, the 16th letter of the sequence including the gap must be A. Now let's see PDB code. By default, Darius loop searches the protein data bank. So it has been connected at the backend with the protein data bank. For suitable loop candidates, it is possible to define a PDB code that will be removed from the search, including its homologs. So homologs filter criteria can be given any, but the approved standard criteria can be greater than 70% sequences identity, so that we can get maximum loop searches for understanding. This is useful for benchmarking purposes. If we want very stringent, accordingly greater than 80 or greater than 90% of sequence identity can be reported. Now what is the pre-configured test for this? There is a demo example, or the demo example has to be kept. So the demo example is CASP11 target T0762, which is an ATMB transfer protein, which has been solved as a PDB code 4Q5T. So this is a demo example which can be utilized, and it is also given in the above server for understanding. The initial homology model was built using modeler, and in our previous courses we have already seen how to do the initial single template modeling and multi-template modeling also. So here you can develop this model through that. The initial homology model will be built through model using a template PDB 2V25. This protein is a glutamine receptor protein which has 90% sequence identity, except an N-terminal motif which comes from PDB 2LK9. So there are two templates for developing this model protein. One is 2V25 and second one is 2LK9, which is a BST2 transmembrane helix. Templates were identified from sequence using HH search method and optimally aligned using TM align algorithm or server. A gapped sequence was defined corresponding to 4 loop regions. So when the sequence was aligned, 4 gap regions were identified, which were recognized as 4 loop regions where the template was missing, extended until all flanks were on helix sheets region of the model. Darius loop workflow. So now let's see the workflow and what results we receive after giving this input as the component. So this server returns 10 models for each loop that is being modeled. One line interactive visualization and model selection facilities are also offered. The main aim of Darius loop is to achieve a high accuracy for at least one of these 10 loop models. In addition, a confidence score is presented. The better the confidence score, the higher the expected accuracy of the best loop. So in the result, if the confidence score is high, the higher accuracy of the loop will be reported. Darius loop does not attempt to predict the best among the 10 loop models. Moreover, in case of multiple loop regions, this server does not attempt to predict which combination of loop model is the best. While such predictions would be very useful, which we have not yet found any algorithm or score or potential that can do this successfully. For example, experimental data such as SAX data could be in a theory to be used to filter the combinations. So now in this loop modeling of Darius loop, let's see section wise. So in the progress report section will incrementally provide information about job progression and errors, if any, whatever the data is been submitted. A typical run should produce a report similar to that. Errors related to the input data specified are now also reported in this section. So now let's see the next section, that is model visualization. Under model visualization of Darius loop, online interactive visualization of the models can be seen, visualized and generated, and which is based on the NGL technique. Different representations can be selected. In this server, a menu makes it possible to select a model among the 10 loop models or to visualize them together. So, by this approach we will be able to represent that. Now, the next one is confidence report. What happens in confidence report, or what we retrieve from there, is this section reports the confidence of loop modeling result among the 10 modeling proteins, which one is the best. For every loop, the confidence score and level are indicated. The better the confidence score, smaller values, the higher the expected accuracy of the best loop. This in the previous lecture also we saw, the lesser the value, the better is the energy minimization. The next one is model archives. So, in that all the loop models will be stored. In this archives, which contains all the 10 models for all the loops in PDB format. It is in the Unix star format, compressed using gzip. Once saved on our computer or instance, tar gz can be converted and all the model proteins can be utilized, seen, or can be opened into another software known as RASMOL. The another result as reported will be model report. Under this section, the reports will include the detail corresponding to the best candidates identified for every loop. For each candidate represents here is protein. We indicate, or it can be indicated, the PDB entry, the chain, the residue numbers corresponding to the fragment. It also gives BC score, flank rigidity, flank root mean square deviation, and different statistical version known as Jensen-Shannon divergence and loop sequence. It also gives the gapped sequence defines loops ranging from different amino acid to different amino acid. So, it gives us the specific regions of the amino acids where the loop is been represented. So, here sometime from 60 to 70, 159 to 84, 259. So, different loops will be reported. In the initial homology models, the loop root mean square deviation will be seen, and after loop modeling, how much the deviation has been reduced or increased will be reported. These values reflect the difficulty or the development of loop model for a specific protein representation. Between the top candidates predicted by the web server, loop candidates will be reported as #1, #2, #3, #4, like that #10 model. Among them, we as an analysis person can select any significant loop model, and which may be selected to see the root mean square deviation as well. So, on the basis of that, fittings and flanks can be reported. For each loop, the results will be represented and the best possible identification or candidate will be shown. This is also may be represented by different figures, as we can see in the presentation, that these specific root mean square deviation, flanked regions and loop models are reported. The another portion of this result will also include analyzing the effect of filtering or CAS11 test sets which we have given. There may be 4 different filtering methods which are sequentially applied in this protocol or in this server. The result of each filter for the loop, specifically for this protein sets, will include like statistical information like sequence similarity, geometric clustering, predicted local conformations, structural clashes and many other which can be reported as the statistical parameters. So, in the figure we can also see the smaller histograms on the top right can be represented and can be seen for these approaches. So, this is the whole pictorial representation and understanding of this server which helps in loop modeling. So, to conclude this whole lecture, we have tried to understand the protein structure prediction by different methods, in which at the starting we reported different softwares and tools for loop modeling and for protein structure prediction. In that, in addition, to enhance our structure prediction and better validation, our approach known as multiple template modeling and loop modeling was also seen in this lecture. We also saw that loop modeling can give us the best fit model after doing this prediction. In this lecture, we also saw a specific server by which we can do loop modeling at our end. For that, input, output and all the result section was being studied. So, in the references we can see this whole presentation and the significant articles by which this presentation has been prepared. Thank you, and now next we will see how for protein validation, for multiple template modeling, for all these approaches, what are the different softwares and another tools for performing these activity. In our coming lecture, we will also see different pipeline for protein modeling. Thank you.